there's a huge amount of stuff to get through today if you looked it over before class like you're supposed to. So I want to get started right away. This is the first of two major lectures on Mendelian population genetics, which is, again, what I've talked about as the sort of product of what's called the modern synthesis. This was from roughly 1920 to 1950-ish, the integration of Mendelian genetics with evolutionary theory, natural selection, and in particular the mathematical description of how genetics of populations works. So the view here is of evolution not at an organismal level, but evolution at the level of genes and gene pools, all of which we'll talk about. So this is kind of, in a sense, redefining evolution in a genetic sense as change in allele frequencies within a population over time. Right? It's the genetical view of evolution by natural selection. So, again, getting away from an organismal way of thinking about things. You tend to think that mice are organisms and mice produce gametes in order to make more mice. Mice reproduce in order to make more mice. You can flip this on its head and say, well, what continues through evolutionary time is genes, and a mouse is a gene's way of making more mice genes in the future. Right? The genes encode a series of developmental stages and adaptations that end up being a mouse, and the function of that mouse is to pass on mouse genes into the next generation. So there's sort of a series of slides here that have a life cycle of an organism where you have adults, what you're used to thinking about as the units of selection as populations of adult organisms. And those organisms produce gametes. The collection of gametes, the collection of genetic variation in populations in sort of haploid state, that's often called a gene pool. It's that reservoir of genetic variation that constitutes the interbreeding population of organisms at the organismal level. So those gametes combine to make zygotes. Those zygotes grow up to become adult population, right? That's kind of life cycle that you all know about, but several things can happen at different stages along that life cycle. So that's what we're going to look at here. So if you have a population of adult organisms in which the frequency of two particular alleles at some locus A, the big A allele has a frequency of 0.6 and the little a allele has a frequency of 0.4. So that's 60% big A, 40% little a, right? If those adults produce gametes without mutation, then you would get this particular collection of gametes, symbolized here as sperm and eggs and big A's and little a's for these different allelic states. And if you really are a detail-oriented person, you could count up the little pictures and convince yourself that it's 60-40, right? Big A. Now, those gametes, haploid gametes, combine to form diploid zygotes. That's through the mating of the adult organisms. In this example here, we're saying if there's random mating, we all know that mating is not random at the organismal level, but what we're talking about is mating random with respect to genotype at this A locus. In other words, if big A alleles are not any more likely to hook up with other big A alleles than they are little a alleles. If they're randomly mating at this locus, then the zygote should be formed in proportion to the allele frequencies in the population. So, if 60% of the sperm are big A and 60% of the eggs are big A, if they combine randomly in proportion to their allele frequencies, you would expect to have 36% big A zygotes formed. We're all good with that? there's going to be a whole series of things involving, 6th to 8th grade math today. So just, you can do it. You can do it all. 40% little a sperm meet with big A eggs with random expectation. That would be 24% of the time, causing this kind of heterozygote. And big A sperm with little a egg, 24% of the time, causing that kind of heterozygote. 0.4 times 0.4 is 0.16. That's the expected frequency of the little a homozygotes. We're all good, right? Okay. So if there was random mating, these are the zygotes that you would get, and they're in proportion to the allele frequencies in that gene pool. And this slide is just the same as the last. It's saying that this frequency should be 36%. It's the product of the allele frequencies. There's two different ways of getting heterozygotes. Big A egg, little a sperm, or big A sperm, little a egg. So these sum together, it's the same genotype to be the 48%. And the little a homozygotes are 16%. These are allele frequencies. What are these? They're genotype frequencies. If you have all of them together, you're accounting for all of the zygotes that are made by that gene pool, what do their frequencies add up to? To 100%. If you add up all of everything, you get 100%. So the sum of the genotype frequencies is 1, or you'd expect it to be 1. So those zygotes grow up to be adults, and if certain conditions prevail as they grow up, if there's no differences in the survival of zygotes based on genotype at the A locus, and if there's no biased migration in or out of the population, biased meaning with respect to genotype at this locus. Right? It's not that there is no migration in and out, it's that there's no one genotype or is not more or less likely to migrate in or out based on genotype at this locus. And if a bunch of them are going to die, but if they die randomly with respect to genotype at this locus, then you're going to get this population of adult organisms. In other words, the frequencies of the adult genotypes is going to be the same as the frequency of the zygote genotypes, if those three things don't interfere. So that adult population of organisms then produces gametes, and they mate. What is the frequencies of the alleles in the gene pool of gametes that those adults produce? Well, you have these homozygotes. What gametes do they produce? Big A homozygotes can only produce big A gametes, right? The little a homozygotes can only produce little a gametes. The heterozygotes produce half of their gametes on average will have the big A allele and half will have the little a allele. So if you add these up, these were 36%. So 36% of that allele plus half of the frequency of the heterozygotes, that's 48. Right? Half of their gametes will be the big A gametes. So that together should be 60% big A alleles. And the little a alleles are produced at half the frequency of heterozygotes plus the frequency of the homozygote little a's. So those end up being 40%. So that's a long way to go. Around one generation of from gene pool to zygotes to adult mice back to a gene pool, and what we have is exactly the same allele frequencies we had before. We started with 60% big A and 40% little a, and we're back at that exact same spot. Nothing changed. Yippee, right? this is, the importance of this is it's a null model. We've specified the conditions under which no evolutionary change occurs. We're defining in the Mendelian population genetic sense, evolution is change in allele frequencies in a population over time. If there's no change in allele frequencies in a population over time, there's no evolution, or at least there's no evolution at this locus. Right? This A locus is unchanged after one generation. So here are the conditions. No mutation. There's random mating with respect to genotype at that locus. No biased migration with respect to genotype at that locus. If there's no selection with respect to genotype at that locus, in other words, the different genotypes have equal fitness. And there's no differential random mortality with respect to genotype, which is called genetic drift. We'll talk more about that on Monday, clarifying what genetic drift is. But if those things happen, then you have no evolutionary change. So that's a generation of mice with big A and little a alleles at the A locus. Now we want to develop a generalized model of allele frequency change over time that would apply to any sets of alleles at any locus that we can have a generalized understanding. So we have, in this example, you have A1 and A2 alleles at some particular locus. And instead of having their frequencies be numeric, 0.6 and 0.4, we're just going to symbolize the frequency of the A1 allele as P. And we're going to symbolize the frequency of the A2 allele as Q. Right? So they apply to any alleles of any whatever their numeric quantity is. And these are the expected genotype frequencies. You should, if the frequency of the A1 allele is P, you should expect the A1 genotype at a frequency of P squared, P times P, etc. So here's again what I warned you about yesterday. You need to make sure you're carefully distinguishing in your head the difference between loci and genes and alleles and genotypes and phenotypes, but also allele frequencies and genotype frequencies, and just keeping straight, doing a little bit of bookkeeping to keep straight the different things that we're talking about here. So if P is the frequency of the A1 allele and Q is the frequency of the A2 allele, I'll underline it as well just for symmetry. If there's only two alleles at this locus, then what's the sum of P and Q? 1, 100%, because their frequencies, they add up to all of them. So if there's two alleles at a locus and I tell you that P equals 0.8, what's Q? 0.2. Sixth grade math is going well. Okay, so here's the general case of recombining gametes. It's exactly the same kind of diagram we did for big A's and little a's with numeric things, but here the frequency of the A1 allele in sperm is P, in egg is P, so this genotype should have a frequency of P squared. Heterozygotes are formed at PQ, except there's also formed at QP. There's two ways of producing these heterozygotes that are equivalent, and the homozygous A2A2 should have a genotype frequency of Q squared. So you have an allele frequency of Q, and you have a genotype frequency of Q squared. Thumbs up? So just, if I say Q, that should tell you that I'm referring to an allele frequency. And if I say Q squared is some number, that should cue you into that I'm talking about a genotype frequency. So now you can just add these up in the same way we did before. You have the genotype frequency of P squared. The alternative homozygote genotype frequency is Q squared. Heterozygotes are at 2PQ because there's two equivalent ways of forming them. those are the genotype frequencies. So we said P plus Q has to equal 1 if there's only 2 alleles at a locus. If you add up all the genotypes, their frequencies add up to 1 as well. So now we have P squared plus 2PQ plus Q squared equals 1 as a statement about genotype frequencies. Just that you've accounted for them all. They add up to the entire population. Okay? So, this null model that I mentioned, the description of no evolutionary change occurring in allele frequencies in a generation, is called Hardy-Weinberg equilibrium. It's called equilibrium because an equilibrium state is a state that is not changing. Right? Something is at equilibrium if it's unchanging. So this was developed independently by two mathematical biologists, Hardy and Weinberg. And basically what this boils down to is if a simple set of assumptions holds, then the allele frequencies in the population will not change over time. And this is mathematically described by having allele frequencies as P and Q, and genotype frequencies as P squared, 2PQ, and Q squared, like we've been building up to. So here are the simple assumptions that are the same as we went through the mouse generation wheel. So no selection. If genotypes survive at equal rates, that means there's no survival selection, and they contribute gametes equally to the next generation, meaning there's no sexual selection. They're all living and breeding equally with respect to genotype at that locus that you're talking about. No mutation. They don't change when the adults are making gametes, or they change in equivalent ratios. There's no net mutational change. No biased migration, right? So genotypes are not more or less likely to enter or exit populations. Again, it doesn't mean that there's no migration. It's just that there's no differential migration in or out of the population with respect to the genotype at the locus that you're talking about. No biased random events. So all kinds of misfortunes happen to organisms, but if they happen randomly with respect to genotype, then on average they're going to take out just as many, well, not just as many, they're going to take out big A alleles in proportion to the frequency of big A alleles. They're going to take out little a alleles in a frequency in proportion to the frequency of those alleles. So you have all these random events happening that kill off a bunch of organisms, but they won't change the allele frequencies if those random events are happening in proportion to the alleles within the population. Okay? Otherwise, it's called genetic drift. Genetic drift is random changes in allele frequencies due to random events. And if mating is random by genotype, so that's the combining of alleles into zygotes, again, we all know that mating isn't random at the organismal level, but if mating is random at the genotypic level with respect to these particular alleles, if they combine in proportion to their frequency in the populations, then you have Hardy-Weinberg equilibrium. You have no evolutionary change. Now, it should be obvious to you as you kind of listen to me prattle on about this that that's total nonsense, right? How often do those conditions hold for populations of organisms in the wild? Essentially never. They might hold for a whole bunch of different loci within organisms, right? If we're talking about evolution of alleles at a locus, you can imagine those conditions applying at the genetic level, perhaps even much of the time. But at the organismal level, that's completely, nonsense, or malarkey, as I say. Those 5 conditions are almost never met, certainly not at the organismal level. But the point is that of this null model is that you can then specify when evolution does occur. So if Hardy-Weinberg equilibrium is a way of specifying when you do not get change in allele frequency, flipping that on its head, it's what allows you to specify what does cause changes in allele frequencies and genotype frequencies in populations. So it is, when I say it's a null model, what I mean by that, it's a random expectation given a specific set of assumptions. Given these assumptions, without departures from this, we would expect no change in allele frequency. So that's the simplistic version of Hardy-Weinberg that you have likely heard before. You shouldn't be surprised to learn that you can apply this to much more realistic kinds of genetics of organisms. I think I mentioned the other day or last week or sometime that this idea that there's two alleles at a locus and that one's dominant and one's recessive is total nonsense. That's kind of a junior high fictionalization of the genetics of organisms. Most loci have lots of different alleles. Does that screw up your ability to calculate and use Hardy-Weinberg equilibrium as a null model? The answer is no. However many alleles there are, you calculate what their frequencies are, the frequency of each of those alleles. If you add them all up, what do they add up to? 1. So here's an example with 3 alleles, with frequencies P1, P2, P3. But you could do this for 20 or 50 or 1,000 or however many you wanted to do. They would all add up to 1 if you've got them all. Then the genotype frequencies are just given as the binomial expansion of their summation. So here, this is drawn out for you with P1 plus P2 plus P3 quantity squared gives you these genotype frequencies. These are the frequencies of the homozygotes here, and these are the frequencies of the heterozygotes here. But hopefully, you can see that all you have to do is expand this out as big as you need it to be. If you had 5 alleles, 10 alleles, however many you have, you should be able to see what the expected genotype frequencies are. We're okay with that? Excellent. Okay. So, these are then the 5 conditions of Hardy-Weinberg equilibrium, and what the sort of 2 population genetics lectures do is we talk about mutation and selection today, and then migration, drift, and non-random mating next lecture. So we're kind of dividing these 5 things that cause violations of Hardy-Weinberg equilibrium into 2 lectures after that introduction. So here's your pop quiz. Since you saw the slides online, it's not really a pop quiz, and I don't care that you don't turn anything in or get any kind of grade. You just have to be able to do this, right? So big A, what's big A? It's an allele. If it has a frequency of 0.9, you might symbolize that as P equals 0.9, right? Does A equal 0.9? No, A is an allele, right? It has a frequency P of 0.9. What's the frequency of the alternative allele, little a, if there's only 2 alleles? Geniuses, each and every one of you. Okay, P squared plus 2PQ plus Q squared equals what? They're going to add up to 1. What are they? What's a descriptor? What does P squared mean? A is an allele. You can't square it. Right. P squared is the frequency of the big A genotype. Genotype. I'm still in 6th grade handwriting, apparently. Okay. So you would then be able to calculate what they are numerically, right? If A has a frequency P of 0.9, what's P squared? 0.81. Whee, it's easy. Okay, so here's sort of a little mini recap. If these 5 assumptions are met, if you describe the frequency of one allele as P, alternative allele as Q,
then the genotype frequencies follow from p squared plus 2pq plus q squared equals 1, and they won't change over time. There'll be no evolution at that locus if these 5 conditions are met, and these 5 conditions, the first of which is mutation. So what we're going to use this null model to do is go through sequentially each of these 5 things and say, if that was the only thing happening in the population at that time, what would be its effect on the population genetics of those organisms, right? So we talked last lecture about mutation being the importance of mutation for the maintenance of genetic variation, the types and classes of mutations that occur. Question now is mutation as an evolutionary force by itself an important, interesting cause of changes within populations of organisms? Does it have a significant evolutionary impact on allele frequencies? So, if you picture the big A allele as the normal functioning protein, the normal functioning gene that causes a normal functioning protein, without which you'd be dead. So a knockout mutation causes you to die, and that we'll call that the little a knockout mutant. You're dead. If the mutation rate from big A to little a is 1 per 10,000, 1 times 10 to the negative 4, which you saw the other day, is on the higher end of estimated per gene mutation rates, 10 to the negative 4, but it's certainly a plausible mutation rate. The reverse mutation from little a to big A is, in this example, considered to be 0, because to get the exact right change, there's lots of ways to knock out an allele and make it non-functional, but to have the reverse change occurs is much less likely. You'd have to get rid of a stop codon or have the frameshift mutation disappear in exactly the right way. So here we're just going to suppose it's 0. So we're making, in our example, we're making mutation as strong an evolutionary force as is plausible. A high but plausible mutation rate with 0 back mutation. And we'll see if the big A allele originally has a frequency of 0.9, how does it change? How would we expect it to change over time? So here's your initial allele frequencies, 0.9 and 0.1. They, mutation converts copies of big A into copies of little a at 1 times 10 to the minus 4 here. Okay. So that's your gene pool. They make these new allele frequencies. You go around here, and has the population changed? That much, right? If you repeat that for thousands of generations, what will be the effect on the allele frequencies? So here, this graph shows, it's for this example, it shows the frequency of the big A allele starting at 0.9, like we said. And over 1,000 generations, because we've said that the reverse mutation is 0, over 1,000 generations it's gone down to, I can't tell you what it is, somewhere around 0.8. So there is some kind of noticeable allele frequency change over many thousands of generations with mutation. So is mutation by itself an evolutionary force? Well, over 1,000 generations, it changed the allele frequency by roughly 10%. So that population at that locus is changing, but boy, is it slow, right? The more interesting thing with mutation as an evolutionary force is when combined with other things, such as selection. So something that you want to have a more sophisticated understanding of is mutation selection balance. It's an evolutionary explanation of why genetically caused diseases persist in populations. One might naively think, oh, if natural selection is so important to evolution, then genetic diseases should
go away. There shouldn't be any. They should have all been eliminated from populations. Mutation selection balance is an evolutionary explanation of why genetic diseases persist in populations. So, we said the other day, mutations occur randomly with respect to fitness. They're not foreseen and forecast as sort of useful things. But if you are an organism that has had a history of selection, your ancestors survived and reproduced, and their ancestors survived and reproduced in a particular environment, it's likely that your genotype is already fairly well adapted to that environment because of selection on your ancestors. So that means that most mutations are likely to be bad because you already have, your lineage already has a history of selection optimizing you for that particular environment. So we call bad mutations deleterious. They lower your fitness. So large mutations are going to almost always be bad. They're very, very, very likely to lower the fitness of the organisms. Smaller mutations, like little changes in amino acid sequence, are much more likely, they're also likely to be bad, but they're more likely to be, have a chance of being beneficial. So the scale of the mutation is related to its likelihood of being positive or negative for organismal fitness. So deleterious mutations are selected against by definition, right? A mutation that is bad for fitness is being selected against because that's what fitness is. But they also get created anew by additional mutations. So the idea of mutation selection balance is an equilibrium state between the rate of mutation and the rate of removal by selection. So if those forces balance each other, then you will have a non-zero equilibrium frequency of a deleterious mutation. Yes?
So big mutations and little mutations are both usually bad, but little mutations are typically more beneficial than big ones? Is that what?
No, they're more likely to be beneficial. So if you would say a small mutation might have a 15% chance of being beneficial, 85% chance of being deleterious. But a large mutation would have a 99% chance of being deleterious. I'm just making numbers up, obviously. But they're, on average, in lineages of organisms that have a history of selection in that particular environment, we expect them to already be well fit to that environment. So any change is likely to make them worse. But big whole-scale sort of changes of the genome are almost always going to be deleterious. It's a probability thing. Okay. So here's a way that you can predict what that equilibrium is. So not only do you no longer have an expectation that selection should eliminate all genetic diseases, you can actually predict how common disease-causing alleles would be in populations. So obviously, if this is a balance between these two opposing forces, the equilibrium condition will depend on the relative strengths of those two forces. If the mutation rate is really, really high, what would you expect the equilibrium allele frequency to be? Right. So you have these two opposing forces. Selection is getting rid of deleterious mutations, and mutation is making new ones. They're in opposition to each other. If mutation rate's really high, so here's mutation rate pushing this way, here's selection pushing this way. If mutation rate is really high relative to selection, what would you expect? Would you expect a lot or not a lot of these deleterious alleles? A lot. If selection is really, really strong pushing this way and the mutation rate's really low, you would expect very, very few of these deleterious alleles. So this Q with a little hat over it, that's the equilibrium, the expected equilibrium frequency of a deleterious allele.
And that expected equilibrium frequency is a function of the mutation rate mu and the selection differential, the difference in fitness between those genotypes S. So even without knowing the units of mutation rate and selection, and even without knowing the details of how that equation works, you ought to be able to see that if S is a big number, then Q hat will become a smaller number. Right? If the mutation rate is a big number, then Q hat will become a bigger number as well, right? Without even knowing the details of what those units are. And that matches your intuitive interpretation of these counterbalanced forces of selection. What about selection? Does mutation cause... So mutation as an evolutionary force can cause allele frequency change over many, many generations acting by itself, but does it cause adaptations? Does it increase the fit of organisms to their environment? No. It's selection that is the non-random part of evolution. It's selection that increases the fit of organisms to their environment. So what this Hardy-Weinberg null model does is it takes as a starting point that all genotypes survive at equal rates. That's survival selection, and contribute gametes equally to the gene pool. That is, they're equally successful at finding mates and reproducing and passing on their genes. So that's a null expectation of no evolutionary change, and selection is when there is differential reproductive success, where some genotypes reproduce more than others. The word differential means not equal. It means that there's differences among genotypes. So here's an example of this kind of crazy mouse wheel. Let's see, where does it start? Oh, it starts with 60-40 allele frequencies. I can hide that. Is that better? 60-40 allele frequencies, they would, if there's random mating by genotype at this locus, you would get those genotypes in those proportions. But suppose 25% of the heterozygotes die and 50% of the homozygous B2 individuals die. That's non-random survivorship with respect to genotype, right? Zero in this example, 0% of the B1 genotype individuals die. So after selection, these are the genotypes of the adults that mate and produce the gametes into the gene pool, and the new allele frequencies after one generation have the B1 allele has gone from a frequency of 60% to a frequency of 67.5% after one generation. Now notice this is really strong selection, right? To have 50% of your homozygous B2 individuals die, that's a very strong selection, 25% of your heterozygotes. But it gets you a huge amount of allele frequency change in one generation. In the plausible mutation example, we had plausible but high mutation rate over 1,000 generations. You had a 10% change in allele frequency. You have in one generation a 10% change in allele frequency with selection, precisely because it's non-random with respect to fitness. Although, as I say, this is an extremely high level of selection. So 7.5% increase in the frequency of the B1 allele, 7.5% decrease in the frequency of the B2 allele, population evolved. This, as I mentioned, is a contrived example with really, really strong selection. This is true for much weaker levels of selection. There's this nice graph where you can compare the frequency of the B1 allele over a number of generations, 1,000 generations, like in the mutation graph,
depending on how strong the selection is. So when selection's strong, this yellow line, you have 100% survival of the B1 genotypes, 90% of heterozygotes, and 80% of the homozygotes B2. And within, I don't know what that is, maybe 25 generations, the B1 allele goes from a frequency near 0 to a frequency that is at 1 within about 25 generations. If the frequency of the B1 allele is 1, what's the frequency of the alternate allele? 0. So the one, an allele that has a frequency of 100% is said to be fixed in the population. It constitutes all of the genetic variation at that particular locus. Now, what I want, that shouldn't surprise you a lot with strong selection. What I want you to notice is this last example, this blue line. Look at the relative fitnesses here. 100% survival of the B1 genotype, 99.8% survival of heterozygotes, and 99.6% survival of the homozygous B2. Though that level of selection is so weak that field biologists could never measure that. The grants going out measuring the beaks of finches would not detect that level of fitness difference. It's just, that is a really, really weak selection. And nonetheless, over 1,000 generations, it causes an allele frequency change that is just as appreciable as the mutation example that you saw. And these intermediate lines of orange and red and green are the courses of selection, how allele frequencies would change with intermediate levels of selection. Did you have a question? It is. The B1 genotype has a systematic advantage over either of the other two genotypes. Systematic advantage, differential reproductive success, that's selection. It's just that systematic advantage is tiny. 100% likelihood of surviving and reproducing versus 99.8. That's a systematic advantage. But over 1,000 generations, it adds up. All right. Here's an example from the book, the ADH gene, which is alcohol dehydrogenase. Fruit flies, these Drosophila melanogaster, what they do is they fly around smelling out rotten fruit, rotten fermenting fruit, and they lay their eggs on that. That's what fruit flies do. That's why they're around your rotten bananas in your house. The rotting fruit produces alcohol, which is why people rot fruit sometimes. This enzyme, alcohol dehydrogenase, breaks down alcohol. So it's in terms of the natural history of these flies, it's a relevant enzyme to their survival because they normally live in an environment that has alcohol. So these guys did an experiment where they had two replicate populations on ethanol-spiked food where the functional alleles of the ADH gene should be relevant. They should matter to fitness. And then they did two control lines on ethanol-free food. So if there's no ethanol on food, what's the relevance of genetic variation in ADH? 0, right? It's not a feature of the environment. And then they just tracked the allele frequencies over time. They did two replicates of each to control for random effects such as genetic drift. They started with the ADHS. The S just means that it was the slow allozyme in the gel electrophoresis. It moved less far through the gel. Started at 65%. In the two control lines, it bounced up and down over time due to random changes, but after 50 generations, it's basically unchanged. In the two experimental lines, E1 and, wow, I should probably say E1 and E2, shouldn't it? But the green and the blue, after 50 generations, this allele
has been selected against, has gone to a frequency near zero. So with the replicated experiment shows you that this is a systematic kind of advantage. So the ADHS allele is being selected against in this particular example. All right. So we know about these 5 assumptions in Hardy-Weinberg and how to calculate genotype frequencies. These are our expected genotype frequencies based on the allele frequencies. Question is, could selection, we know obviously selection can change allele frequencies. That's what we just did. But does selection change genotype frequencies in a way that is important evolutionarily? And the answer is it can, but if there's random mating, that it gets restored after one generation. So what we'll talk about is a case of heterozygote advantage. Can anyone imagine what heterozygote advantage means? Who does the best? Go way out on a limb here. Is it probably the heterozygotes? Again, little geniuses. All right, so here's this mouse wheel example where you have, if you're starting with equal allele frequencies, so P equals Q equals 0.5, they're both at 0.5, you get these genotypes, and the survival selection here is that you have 50% of each of the homozygote genotypes dying, but the heterozygotes survive just fine. So the heterozygotes do better than either of the homozygotes. That's why it's heterozygote advantage. Right? So here's the genotype frequencies after selection. You can see if you found a population of organisms and you said, well, the B1 allele is at 0.5 and the B2 allele is 0.5, you would expect to have these genotype frequencies. But if you actually genotyped your organisms and found out that they had these genotype frequencies instead, you would say this population of organisms is not in Hardy-Weinberg equilibrium. These are not the genotype frequencies you would expect to get based on those allele frequencies. But what happens when these guys reproduce? When those guys reproduce, there are equivalent numbers of each genotype. There's been no selection on allele frequency. These died equally, equally here and here. So when these guys make gametes, the allele frequencies are actually unchanged. So you have selection that is causing genotype frequencies to deviate of adults, to deviate from Hardy-Weinberg expectations without causing changes in allele frequencies. We're going to talk more about heterozygote and advantage and disadvantage in a minute, so we'll kind of get back to this. All right. So when we developed Hardy-Weinberg case for alleles, we started out with a special case of big A and little a with some particular numeric kinds of allele frequencies. And then from that, we built a general model where we symbolized the frequency of the big A allele as P and the frequency of the little a allele as Q. And we developed this general model, P plus Q equals 1, P squared plus 2PQ plus Q squared equals 1. Question is, could we do an analogous thing with a model for selection? So, last week, or whenever it was, I said selection acts on phenotypes. And that indirectly causes changes in genotypes. Phenotypes are what they are because of the genotypes that underlie them. So when we're talking about, in the world of population genetics, where we're focusing on the gene level, not the organismal level, we're going to talk about genotypic fitnesses, the fitness of a genotype. But don't get confused. Selection is acting on that genotype only through its effect on phenotype. And it's the effect of the phenotype of the whole organism. But we're simplifying to the genotype at a locus for this example. We're just pretending that there's 100% direct correspondence between genotype and phenotype, and when selection acts on the phenotype, it is affecting genotypic fitness. So, fitness is, in evolutionary biology, symbolized as this, in my font it's a W, but it's actually an omega. Okay? And the fitness of a genotype is symbolized with subscripts. So the fitness of the A1 genotype, its fitness is symbolized as, W, I'll just call it W, W11. The fitness of the heterozygous genotype is W12. And the fitness of the A2 genotype is W22, not surprisingly. So, the question is, can we develop a predictive model of how allele frequencies will change over time dependent on their relative fitnesses? So, if there is selection, selection means unequal fitness. But you need to standardize that unequal fitness relative to something. And what it is standardized to is the average fitness of all genotypes. So, W with a little bar over it is average fitness or mean fitness. Well, how do you figure out the average of anything? How do I figure out the average of 10 numbers? I add them all up and divide by the numbers of observations. If I'm dealing with frequencies, I've already divided by the numbers of observations. That's how I made them into frequencies, right? So average fitness, mean fitness, equals, what's that? P squared. P squared is the frequency of the A1 genotype. So we're already, it's a frequency, we're already dividing by the total there. So mean fitness is the frequency of this genotype times its fitness, plus the frequency of this genotype times its fitness, plus the frequency of this genotype times its fitness. You're adding them all up, dividing by the total, but because they're already expressed as frequencies, there's no divisor that you see. There really is one, but it's implicit when you made these as frequencies. So that's what mean fitness is. Here's the relative fitnesses of these different genotypes. So here's the frequency of this genotype times its fitness divided by average fitness, etc. So far, so good? We might have got a little out of 6th grade math, and we're headed towards 7th grade math, 8th grade math. I don't know. Well, you're okay, right? Give me a... Yeah. If we want to compare the fitness of these different genotypes, we need a common standard to compare them to. So that common standard is mean fitness. They're all being divided by the same thing. And this mean fitness is calculated based on the fitnesses of each of those genotypes. I'll go back a slide. So mean fitness comes from the collection of all of them. And so then each of them is expressed relative to that metric, which reflects how they all do collectively. So it's just a way to standardize the relative fitness of a genotype. Okay? So, can we describe in a way that quantitatively predicts how allele frequencies change over time? So if we want to know, for example, how many A1 gametes are produced, well, it's going to depend on how many A1 individuals there are in the population and the fitness of the A1 genotype. and it's also going to depend on how many heterozygotes there are and the fitness of heterozygotes. Right? A1 gametes are produced by A1 homozygotes exclusively and by heterozygotes. How many of these alleles are produced depends on how many of those individuals there are and what their fitnesses are. So here's the frequency of individuals that produce only A1 gametes. And here's their fitness. PQ. PQ is half of 2PQ because heterozygotes produce A1 and A2 gametes. Half of their gametes are the ones we're interested in. So the frequency PQ is half of the frequency of heterozygotes, which is the frequency of production. Well, it's not the frequency of production. It's how many of those A1 gametes are, in a sense, locked up in heterozygotes. And their fitness is how good they are at transmitting those to future generations. So that's the production of the A1 allele into the gene pool. Similarly, you can say, well, what's the production of the A2 allele into the gene pool? This is exactly the same thing. Half of the gametes produced by heterozygotes are the A2 allele, and all of the gametes produced by the A2 homozygotes are A2 gametes. The only reason to do it for A1 and A2 is so that you can see later that you could actually generalize this. If there were 10 alleles, you could do this as well, right? If there's only 2 alleles, you can do just one and then calculate the other by subtraction. But regardless. Okay, so how does allele frequency change? Well, delta P is change in allele frequency. The change in something is the new allele frequency minus the old allele frequency. Well, we just figured out that that's the new allele frequency. That's how many of these A1 alleles are produced. It's the frequency of that genotype times its fitness plus half the frequency of heterozygotes times their fitness divided by average fitness. You can ignore all this other crap on the slide. Just skip that. If you stop here and understand through there, then you're okay. So I want you to understand what delta P is, what it symbolizes, but also what it means logically and how it's calculated. It's the frequency of the new allele frequency minus the old is the change in allele frequencies. And you should understand how that new allele frequency is derived, where that comes from. So, we did it. Woohoo! We have made a general expression that quantitatively describes how allele frequencies change depending on the average fitness of that allele. Now we're doing really weird things with language. Now we're talking about not just the fitness of a genotype, even though we know selection acts on phenotypes. We're now talking about the fitness of an allele when it combines with others. Again, you're working at this at the genetic level, not the organismal level, is why we have such weird language about it. If there's 2 alleles, if delta P is plus 0.2, and there's only 2 alleles, what's delta Q? Minus 0.2, right? Because they still add up to all those alleles. You could figure out delta Q this horrendous way, but if there's only 2 alleles, then an increase in the A1 allele has to be matched by a decrease in the A2 allele frequency. The only reason to spell it out like this is you could have 10 alleles, and then you could figure out delta P, delta Q, delta R, delta S, delta T, however many alleles you had. And you could work it all out and be very happy with your answer. So, now we have these allele frequencies, we have average fitness, we have an expectation of being able to predict how allele frequency should change based on the relative fitness of those genotypes. So if we know how strong selection is on a particular genotype, you can predict what the allele frequency change should be. That sounds pretty good. I mean, that's kind of the hallmark of science, is being able to dissect the world into a way that allows you to make predictions about it that you can then verify or not, right? Being able to predict allele frequency change in some kind of quantitative manner, depending on the fitnesses of those genotypes, is a pretty neat thing to be able to do. You can all do it in your spare time at parties this weekend. So, the next topic is patterns of selection and how they differ, how we would expect allele frequency change to vary depending on things like if the allele is, if an allele is beneficial, we expect it to increase in frequency by definition. If it's deleterious, we expect it to decrease in frequency by definition. But does it matter whether they're recessive or dominant, for example? Does it matter what if heterozygotes have different fitnesses from either the homozygotes? They might have higher or lower. And then we'll talk about frequency-dependent selection, which is an entirely different way of conceptualizing these fitnesses. So you all learned whenever you learned this, dominant recessive alleles. In intro bio for a while, we pretend that there's two alleles at a locus and one's dominant to the other, right? So big A is dominant to little a if big A genotype equals the big A phenotype, and the big A, little a genotype also equals the big A phenotype. You all remember that from times past. And the alternative, you only get the little a phenotype if these two recessive alleles are combined into the same genotype. You all remember that. So, if the fitness of the homozygous recessive genotype, the fitness of little a equals 0, that would be a lethal homozygous recessive disease. How do the frequencies of that little a allele change over time if it's common or if it's rare, dominant, recessive, etc.? So the thing you have to realize is that if the little a allele is common, selection doesn't work on alleles. It doesn't even directly work on genotypes. It works on phenotypes. So if the little a allele is common, how common will be the homozygous recessive genotype? Well, let's just give if the little a allele has a frequency of 0.8, what's your expectation of the frequency of the homozygous recessive genotype? 0.64. The higher the frequency of the little a allele, the higher the expected frequency of the homozygous genotype. So if little a is pretty common, then the genotype AA is also pretty common. That's what gets exposed to selection. Selection doesn't see little a's and big A's, right? It sees phenotypes that are combined into organisms. So selection isn't working against the little a allele, it's working against the phenotype produced by the little a genotype. Selection has no effect on, in the perfect example, these heterozygotes have the fitness that is the same as these guys, right? If this is a perfectly recessive, here's this lethal homozygous recessive allele, the fitness of heterozygotes is the same as the fitness of homozygous dominant individuals. So you can see selection is not working against that allele in that case. It's only working when they're combined into homozygous genotypes. So, if your starting conditions have this little a allele is pretty common, if it starts out as common, then Q is a pretty big number, so Q squared is a pretty big number. So selection is, there's lots of sick, dying individuals. Selection is getting rid of that allele very, very quickly. You'd expect it to decrease really, really quickly. If as it decreases in the population, it's becoming more and more rare. So Q is becoming a smaller and smaller number, so Q squared is becoming a even smaller and smaller number. So the numbers of individuals that are getting sick and dying of this disease, starts to approach, get fewer and fewer and fewer, it starts to approach zero, which means that selection against that allele is approaching zero. Almost all of the deleterious alleles are in heterozygotes, not in homozygotes, is what you would expect. So here's a really cool experiment with flower beetles. It was started with 100% heterozygotes. The reason for that was that's a simple way to know that the initial allele frequencies are both 0.5. So P equals Q equals 0.5, and what was started with these flower beetles was a deleterious recessive allele where the fitness of the homozygous recessives was 0. So over 12 generations, what you see is the lethal recessive allele starts at 0.5, it drops sharply, and then flattens out over time. So what's delta P over here? I don't mean what it is as a number. What's delta P over here relative to delta P over here? There's a lot of change really rapidly here because the allele, this lethal allele is pretty common. Yeah, there's a lot, that delta P is a bigger number over here than it is over there. Even though the selection hasn't changed, right? It's still this lethal recessive. It's just that as this lethal recessive becomes rare, very, very few individuals get the disease, so don't develop the phenotype, so they're not subject to selection. There's 2 alleles, so the other one goes up in frequency, right? So this is just the inverse of that. So the rate of evolutionary change with dominant and recessive alleles depends on the frequency of those alleles in these initial starting conditions. But as these deleterious recessives become rare, the selection against them becomes less and less. So it's nice. Data agree with our prediction. Here's the actual models of the frequency of these different alleles. And the point of this graph is you can see and track this already. You can see that the deleterious recessive is going to go away pretty quickly. Well, it decreases in frequency pretty quickly, but then stabilizes at it will go away eventually. If selection is the only thing happening, then eventually this will get to 0. But it's approaching it at an asymptote. It's a very, very slow, gradual approach to 0. What is also shown on here is this other graph on the side, mean fitness, right? W bar. What happens to mean fitness of the population as the frequency of the beneficial allele increases? It goes up and goes up and goes up, right? Shouldn't be a surprise to you at all. That makes wonderful sense. Okay, if there's selection for a recessive, now the recessive is good for fitness. But suppose that recessive allele is at low frequency. How many individuals get the benefit? Very, very few. So what's the rate at which it increases in frequency in the population? Really, really slow is what we ought to expect. Then as it starts to increase in frequency to a point where more and more of individuals are homozygous recessive, you should start to see rapid changes in the increase in frequency of that beneficial allele. And that's what this graph is in the case of these particular fitness functions, 0.4, 0.4, and 1. But it just makes the point that it's a very, very slow, gradual increase, and then kind of a sudden threshold effect, what's the equilibrium frequency of this beneficial allele? So again, equilibrium means the unchanging, constant, steady state. It's 1. It takes a long time to get there, but if selection is the only evolutionary force, and we're going through Hardy-Weinberg, right? We have these 5 things. If there's not mutation, if there's random mating, if there's not migration, there's not genetic drift. If selection is the only thing happening, the equilibrium frequency of a beneficial allele is always 1. The equilibrium, or fixation, the equilibrium frequency of a deleterious allele is always 0. Okay. Oops. All right. Next kind of topic is heterozygotes having intermediate fitnesses. So dominant and recessive means that heterozygotes are the same as the dominant phenotype. That's typically not the case, despite what you learned in intro bio or whatever. Heterozygotes can have fitness that is intermediate between the 2 classes of homozygotes, in which case that changes the rate of evolution, not the outcome. So I just told you what's the outcome, what's the equilibrium state, equilibrium frequency of a beneficial allele. I just asked you that, and you told me it's 1, fixation. Right? If selection is the only thing happening, a beneficial allele will go to fixation. If heterozygotes have fitness that is intermediate, that's still true. The equilibrium frequency of the beneficial allele is still 1. If the heterozygotes have fitness that is higher than either of the homozygote classes, that's called overdominance. And if the heterozygotes have fitness that is lower than both of the homozygote genotypes, that's called underdominance. And in these cases, you get different evolutionary outcomes of the allele frequencies. So here's an example with overdominance. These are the genotypic fitnesses. The fitness of the big A genotype is 73.5% of the fitness of the heterozygotes. So the highest fitness genotype has a fitness of 1. The fitness of the little a genotype is 0. So that's that's lethal to get 2 copies of this allele. But guess what? If you have 1 copy, your fitness is higher than if you have 0 copies. Right? So this is overdominance. The best genotype to have is the heterozygote genotype, in which case the equilibrium frequency of this allele is some number greater than 0. Most people would look at the genetics of this and say, oh, this is a deleterious allele. Here's something that if you get in 2 copies, you're dead. Most people say, oh, that's a disease-causing allele, it should go away from the population. But you don't actually expect it to. You expect it to have an equilibrium frequency greater than 0 because heterozygotes do better than anybody else. All right, so here's this example in these Drosophila where they have these 2 populations that they different starting conditions. They let it run. They don't know what the fitness affects. They don't know, in this particular example, why heterozygotes do better, but the equilibrium frequency is somewhere around 0.8. In other words, if you start the population down here, it goes up, the frequency of the allele goes up and converges at around 0.8. If you start it higher than that, 0.98 or whatever that is, it goes down and converges towards this intermediate allele frequency. Is that a beneficial allele or a deleterious allele? It's neither one, really, right? It depends on, your fitness depends on your genotype. This is a lot like the case of sickle cell anemia that we mentioned the other day, where in environments where malaria is prevalent, heterozygotes have higher fitness than anybody else. So you expect the sickle cell allele to persist in the population at a non-zero number because of its fitness effects in heterozygotes. Okay, here's a generalization, a generalized model of overdominance. So again, you have mean fitness, W bar, as a function of P, as a function of the frequency of one of the alleles. If heterozygotes have highest fitness, then when does the population have highest average fitness? When there's a bunch of heterozygotes. When do you have a bunch of heterozygotes? When both alleles are still present in the population. So if P is a really small number, you don't have a lot of heterozygotes. Average fitness is kind of low. If P is a really big number, that means Q is a really low number, you don't have a lot of heterozygotes. So you have highest fitness when P is an intermediate number. When would you have highest possible fitness? If heterozygotes do better than everybody else, what's the best, the highest possible mean fitness? Or not as a number, but what's the condition that gives you highest possible mean fitness? If everyone was a heterozygote, which you would get if P equals Q equals 0.5. That gives you more heterozygotes than any other allele frequencies. Okay. Underdominance. What if the heterozygotes have lower fitness? Here the answer is a lot more complicated. And there are equilibrium conditions. There are stable outcomes. But what equilibrium condition you get depends on the starting allele frequencies and if there are little random departures from those starting allele frequencies. So one allele is going to go to fixation, 100% frequency, and the other one's going to be lost with underdominance, but you don't necessarily know right away which one. So with overdominance, you know both alleles are going to be maintained in the population at some intermediate equilibrium. With underdominance, you know that one's going to go away and the other one's going to stay, but it's harder to figure out which. So here's a fitness surface. Average fitness as a function of for P for underdominance. So obviously you have, if heterozygotes do the worst, you have highest average fitness if there aren't any heterozygotes. So you have high fitness if one allele or the other prevails in the population, but not both. So this kind of thing is sometimes called an adaptive landscape. So here are these peaks. It's like a mountain range with a fitness peak here and a fitness peak here, but here's a low fitness area here. So that's why it's called a landscape. Oops. Get rid of all that mess.
What does it mean for the fitness to be above 1?
Oh, in this particular graph? I'm not sure. They're usually standardized to the highest fitness. That's a good question. I don't know. I'll try to figure it out. Okay. Oh, I'm going to go back to this adaptive landscape. So if this is kind of going to be an analogy